Okay, let's get started. So welcome everyone. Today I want to walk you through a comparison that I think is increasingly relevant for anyone is doing the computational biology and AI-driven biomedical science research. This is to compare the Biomni Lab AI agent versus Claude Science AI agent. So these two of the most capable AI platforms for biomedical research right now, and they are often mentioned in the same breeze, but they were designed with fundamentally different philosophies. At the end of this talk, you will have a clear picture of what each platform does best, who should use which, and also perhaps most interestingly, how they are actually related to each other and a very deep level. So we will cover like dimensions of the comparison, definition, philosophy, workflow, and functionalities, tools, applications, use cases, users, and also the connections and so on and so forth. This is the dimensions that we will cover in the next slide. Then we will also look at a real-world case study that reveals something surprising about the relationship between these two platforms. Okay, so let's dive into it. So let's start with some basics. On the left, you can see of this slide, there is a identification table, and on the right is our first comparison figure. Biomedical Lab comes out of the Stanford University and Fellow. This it is primarily targeted to be an AI biomedical scientist, not just a tool, but an autonomous agent that can do biology. So its core idea is biology first. It starts with the biological problem and works outward. For the Claude Science from the Arabic, it takes a different angle. It is an AI scientific workbench. Think of it as an integrated research environment where you can do everything from the reading papers to submitting the HPC jobs to drafting manuscripts, and also for the downstream and translation, and also the drug development, all in one place. So this kind of two platforms comparison captures the difference well. So basically for Biomedical, it is an AI bioinformatician that knows how to use biology tools. For the Claude Science, it is more like a VS Code plus Jupyter Notebook plus Biomed and plus HPC in one scientific IDE. So same broad domain, AI for science, but very different entry point with two AI platforms. So keep these two kind of like difference in mind as we go deeper. So the deepest difference between these two platforms is the philosophy. It is about what questions they are designed to answer. Biomedical Lab asks, how do I solve this kind of biology problem? When you give it a task, say, analyze it. And for example, is to analyze the single-cell RNA sequencing data set or predict this protein structure. It automatically decides which tools to use, which database to query, which pipelines to develop and to run. You don't need to know whether to use the ScanPy or any other tools. Biomedical fix that out. For the Claude Science, it asks us a different question. How do I conduct this entire research project? So it is not just a saving one analytic task. It is managing the full arc from the literature review through the manuscript submission with reproducibility and prevalence built in at every stage. So neither approach is better in one's absolute terms. They're optimized for different moments in the research life cycle. For the Biomedical, it excels in when you have a specific biological question and analysis required to run. But for the Claude Science, it is good at when you are managing a multi-week or long-term research project with many moving parts. So this is philosophical split drives almost every other difference we can see for today's lecture. So let's look at how each platform actually works step by step. Okay, and the Biomedical workflow actually is tight and the biological focused. You start with a question, and then it finds the right biological tools, and then it will plan the analysis, runs the pipeline. So actually, before it runs the pipelines, it will using the Python or R, which you can customize to develop the pipeline or the analysis workflows, and then it will validate the pipeline and run the pipeline. So it is well generated integrated running platforms with the final output and results. 5 steps in total for the Biomni, and it is highly automated and also biological specific at every stage. For the Claude Science, its workflows is more like a broad scientific research work. You need to define the question, and also you can do some literature review and planning and do some coding. You can also execute the your research jobs in the high performance computing platforms. Finally, it will visualize and it writes the results for you. And also there is some review agent in the Claude Science which will check the work. So 8 steps in total for the Claude Science. For the Claude Science, which to conduct the full research life cycle. I want to notify you that the Biomedical 5 steps maps roughly onto steps number 3 through 5 of the Claude Science workflows. That is not a coincidence. Biomedical actually is optimized for the analytical core of the research, while the Claude Science wraps the core in a full project management layer. So that's the reason I want to look to you that the Biomedical's 5 steps actually maps the 3 of the 5 steps of the Claude Science. Okay, so for the bioinformatician who just need to run a GWAS or annotated genome, so Biomedical 5 steps workflow is exactly what you want. It is faster, focused, and no overhead. For PI managing a multi-month study, Claude Science 8-step workflow gives you the full picture and also the full landscape of how you need to manage your full project within these 8 steps. Okay, so that's the workflow comparison for the Biomedical Lab versus Claude Science. And this table gives you a quick read on where each platform is strong and partial or limited access across 14 core capabilities. So the pattern is quite clear when you look at this functionality matrix. Biomedical dominates the biological specific capabilities, running pipelines, selecting the biomedical science database automatically, and calling the specialized software, designing the wet lab experiments based on the experimental protocol. They can also integrate and process multimodality data. These are the core strengths of Biomedical Lab. For the Claude Science, it dominates the research life cycle capabilities, start from the literature retrieval, HPC job execution, figure creation, manuscript drafting, provenance tracking, and workflow orchestration, cross-domain collaboration, and so on and so forth. So that's the core part of the Claude Science. Both platforms are generate Pythons and R code to retrieve the literature and also propose hypotheses and implement the Python pipelines. So I think that's the shared foundation between the Biomedical Lab and also the Claude Science. The limited and the partial interests are honestly assessment. Neither platform is trying to be everything. So Biomedical does not pretend to be a manuscript actor, right? But for the Claude Science, that doesn't pretend to be a specialized bioinformatics pipeline runner. So knowing this kind of boundaries help you choose this right tool for the right task. And for the tool ecosystems, for the Biomini, it has very high. It has very high depth. And if you look at the tool ecosystem of the Biomini, it connects around 150 tools and 59 biomedical database and 106 software packages, all curated from over the 2,500 papers which the Biomini Lab collected. So that's not a list of someone assembled arbitrarily. It represents the actual computational toolkits and that the biomedical research community use very often. When you give the Biomini Lab a task, it automatically selects from this kind of ecosystem, and you don't need to know whether to use BLAST or Diamond for sequence search, or whether to use a STAR or the HI-SET2 for the RNA sequence alignment. Biomini Lab decided. For the Claude Science, it takes a connector-based approach. It links to an authoritative scientific resource like the publication mat, and UniProt or the protein database, and also the benchling or the 10 times genomics, right? For this kind of connections using the API or any other MCP connections. So the key differentiator here is the permanence. Claude Science tracks exactly what was run, when, and with what parameters, which is critical for the reproducibility. Both approaches are valid from my point of view and from my personal experience, but mainly optimized for biological depth. Claude Science optimized for the scientific rigor and auditability. Okay. So this table actually makes a depth versus breadth trade-off for a very concrete way. So Biomedical is left core in 6 biology specific domains: genomics, proteomics, and microbiology, pharmacology, structurology, single-cell analysis. These are the domains that I think it has very deep and specialized tooling. For the Claude Science, it's core in 6 research lifecycle domains, like the literature review, research planning, HPC, ultrasaturation, scientific writing, reproducibility, and cross-domain validation and collaboration. Notice this kind of symmetry, 6 and 6, right? And this isn't a coincidence. I think it exactly reflects the complementary design philosophies that we were discussed earlier slides. Both platforms, I think, support the other cross domain at a supported or partial level, so they're not blind to each other's territory. But if you need a deeper genomics analysis, I believe that the Biomini Lab is the right choice. If you need to manage a reproducible multi-domain research project, Claude Science is the right choice. For many researchers, the answer is use both. So that's my suggestions, okay, for you. So let's get concrete a little bit. And here are 9 tasks where Biomini Lab is clear the choice. For example, the variant interpretation, Biomini can take a VCF file and automatically annotate the variants with the functional effects or clinical significance or the pathology predictions. For the protein structure prediction and CRISPR guide design, molecular docking, and drug repurposing, these are all the tasks that are required, which specialized tools that the Biomini Lab can use and how to run it in the Biomini. Biomini knows this kind of tasks. So 2 that stand out, I want to point to you is one is the rare disease diagnosis support, where Biomini Lab can synthesize genomic, clinic, and also literature evidence to support the diagnostic reasoning, and also the wet lab protocol generation. And so the Biomini Lab can design the detailed experiment procedures that have been validated as equivalent to the expert level work. So that's 2 points that I want to point out for the Biomini Lab, which is very promising. So these are the tasks that we're having like 150 specialized Biomini labs tools that you can dispose, and also the intelligence to choose one of them. So which makes a very decisive difference. And now for the flip side, and the A task as well, Claude Science is the clear choice. Number one is systematic scientific literature reviews, which Claude Science can search the PubMed, retrieve papers, synthesize findings, and produce structured reviews with citations. And also the second one is multi-step research planning, and the third part is reproducible analysis. Every step is tracked with prevalence, so you can reproduce very detailed results within months. HPC job orchestration, and it can also help your submit and monitor and retrieval results from computer clusters, which is essential for the large-scale genomics works. And there are also some other works that Claude Science excels in, for example, manuscript drafting, cross-domain scientific orchestration, and large pre-provenance tracked workflows. So for the large and auditable workflows, I think if you are running an analysis that needs to pass peer reviews for regulatory scrutinies, Claude Science provenance tracking is a good choice for you. So notice that this kind of use cases span entire research life cycle, not just an analytic biology case. So let me give you a practical decision guide. So if you are a wet lab biologist who is managing a research project from start to finish, reading papers, planning experiments, analyze data, writing up the results, I believe Claude Science is your platform. It is designed for the end-to-end workflow. If you are a bioinformatician or computational biologist or computational AI scientist for biology, biomedical science, who needs to run specific pipelines, for example, the GWAS or the single-cell RNA sequencing, protein structure prediction, and annotation, variant calling. So I believe that Biomni Lab is your platforms. It really has some specialized tools that you need to use. If you are an AI researcher focused on multi-agent systems, reproducibility, and workflow orchestrations, Claude Science, the architecture is more relevant. Okay. So bottom line from my point of view is like for most researchers, the optimal strategy is to use Biomedi for analytical core and Claude Science as the research life cycle wrapper around it. So which brings us to something, which really brings us to something surprising. Computational infrastructure is often a bottleneck in the modern biomedical research. So let's look at how each platform handles this the computational infrastructure. Biomedical Lab compute every stage, and for the computer story, and the Biomedical Lab actually centers on the Claude's 200,000 token context window. This is a game changer for the biology. A single genomics experiment can produce thousands of files. Literature research spans millions of papers, right? So most of AI models truncate this data and lose critical connections. Biomedical Lab, powered by the Claude, actually can process entire genomic analysis, multi-gigabyte dataset or 1,000-page studies in one single context. That's what enables 800. That's the reason it enables like around 800 times speed up with the power from the Claude. So we will see this kind of progress in the future for sure. For the Claude Science computational story, it centers on the HPC orchestration and provenance. It can submit jobs to the computer cluster, monitor them, and retrieve results and track every step with full provenance. So NVIDIA Biomedical, I think Yeah, the NVIDIA Limo connector gives it access to the GPU accelerated biology models from the Claude Science. So this kind of connection from the NVIDIA platform to the Claude Science is very, I think, is very exciting, and it can run a parallel multi-agent workflows, multiple analysis simultaneously in one cluster. I think the key difference between the Biomedical Lab and also the Claude Science regarding the computational resource is that the biological depths within single powerful context window. Claude Science optimized for distributed, reproducible computation across the HPC infrastructure. Okay. So despite this kind of difference, Biomedical Lab and Claude Science shared substantial common foundations from my point of view. 7 core capabilities that both platforms provided in the same way. Both use the language model reasoning as their core. Both generated Python and R code. Both retrieve static literature. Both integrate external biology tools. Both support the multi-step planning and automation, and they can also generate the publication-ready outputs. Both can propose hypotheses. So I think this shared foundation is why these two platforms are complementary and rather than the competitive, because they are not fighting over the same territories. They are building on the same foundation in the different directions. So this convergence trend is real and accelerating. Biomedical Lab is expanding from the specialized biology agent into a full integrated research environment. But for the Claude Science, it is adding more biological specific capabilities and connectors with each release. I believe that in the 2 or 3 years, the distinction between these two platforms will maybe much less sharper than it is today. But right now, the difference are meaningful enough to guide your platform choice. Okay, so which brings us to the most interesting and exciting part of this talk. And now for the part that rephrase everything we have discussed. And this is the real case study published by the Asrobic. You can find it at the Claude customers Biomni. This kind of blog post. So the problem is like Stanford universities behind Biomni watched their colleagues spending 80% of their time on repetitive tasks. Like for example, searching the literature review, processing data, and adapting protocols. The critical insight will be buried in an unexplored dataset simply because humans could not process the volume. They did more than incremental increments. So the results are exciting from this kind of slide. You can see that Biomedical Lab, which completes the variable bioinformatics analysis in 35 minutes versus 3 weeks for human experts. And that's 800 times faster on that specific task, and 100 times faster overall across the biomedical discovery workflows. It achieved human-level performance on the lab bench and also the sequence QA benchmarks. It designs cloning experiments validated as equivalent to a 5+ year expert in the blind blind testing. It automates the joint sequence RNA, single cell sequencing, joint single cell RNA sequencing analysis, and it also reaches state-of-the-art performance on the HLE exam and 8 biomedical tasks. So these are not incremental improvements. They are order of the magnitude changes for this case. So after extensive evaluation of the AI models, the Biomedical Lab choose Claude for the specific 3 specific reasons. So this is an important context for understanding the Biomedical Lab and the Claude relationship. First, the deep biology knowledge. When the team tested identical tasks across different models, Claude showed a superior understanding of the biological concepts. So as the founder of the Biomni Lab machine put it, it clearly has extensive biology knowledge embedded in this training. Okay, so this is just not. It is not just about knowing the biology facts. It's about reasoning correctly about the biological system. Okay, and the second part is the 200,000 token context window. So it has proved essential for handling the massive scale of the biomedical data. While other models truncated this kind of information sometimes and lost lots of critical details. For the Claude, it can process the entire genomic analysis and a multi-gigabyte dataset and 1,000-page studies without missing the crucial connections. For biology, well, context I think is everything. That's everything. That's for sure. And this really matters a lot. For the third reason, why Biomni team selected Claude is that the scientific reasoning capabilities, synthesizing the findings across the discipline, generating test table hypotheses, identify the patterns that a human researcher might miss, and this kind of compilation of knowledge, capacity, and reasoning mode made the Claude the clear choice for the Biomni Lab. So the code at the bottom actually captures that the Claude demonstrated the best performance across our benchmarks, and particularly in the scientific and the biological knowledge, coding ability, and agentic workflows from the Biomni Lab team. So this slide shows the architecture diagram on the left side of this slide, and it is actually a layered stack of the architecture design of the how Claude powers Biomedical. So I think this is a key insight. Biomedical Lab is not a competitor to the Claude, and it's actually its own application built on the Claude models, large models. Claude provides a foundation model capability, and then the Biomedical Lab provides a biological specific agent layer and a tool ecosystem. The 5 core architectural capabilities make this concrete. And unified environment access, which is to connect 150 tools, 59 database, and 109 pages curated from the 2,500 papers. And the second one is dynamic workflow composition, which creates the input specific plans on the fly. And also there's no predefined templates, and a multimodal data ecosystem, which is very promising to handle the genomic data, proteomic data, image data, and clinical data simultaneously, right? And also the protocol design generation, which is to design the experiment procedure that match the export standards. And finally, it's cross-domain synthesis, which is to identify the connections between the disparate research areas and to generate some innovative hypotheses. I believe that the validation result is really remarkable because it clones the experiments designed by Biomedical Lab, which will be validated as equivalent to a 5+ year expert in the blind testings. So this is not just a benchmark; it's a real-world expert evaluation and validation from the Biomedical Lab. What's the impact on the future of the AI augmented science? The impact of this kind of collaboration really extend well beyond the time savings for the collaboration between the Biomedical Lab and also the Claude company. Yeah. Okay. So the first one, you can see that the real world impact is from the democratization effect. It's very profound. Before the Biomedical Lab, a resource-limited lab cannot compete with a well-funded institution that had a team of bioinformaticians. Now, a single researcher with access to Biomedical Lab can pursue much more research threads that previously required the entire teams. So researchers compete on ideas, not on the computational resource. And also the 100 times speed up changes what's what is economically feasible. Hypothesis that would have taken months of setup, right? Now can be tested in hours with the Biomni Lab. And one team discovered lower transcription factors regulating the lineage findings that are emerging from the Biomni Lab's comprehensive analysis of a massive single-cell dataset that a low human team can have processed in a reasonable time frame. So this kind of flexible autonomy model is important for the biology scientists because they can choose how much control they can retain from the copilot mode to the fully automated execution. This isn't just about represent the researchers, it's about amplify the capabilities of the biological researchers. And the Biomedical Lab, I think now is free access to the scientists worldwide at the website. The collaboration with the Anthropic has made cutting-edge AI-driven biology available to any researchers with an internet connection. Okay. So let me close with these 5 key takeaways that you can act on. And first, I believe that the Biomedical Lab and the Claude Science are complementary AI platforms for biomedical science. They are not competitive to each other. So use Biomedical Lab for the biological specific analysis. For example, the GWAS single-cell RNA sequencing, protein prediction, and CRISPR design. And you can also, and then you can use Claude Science for the research life cycle orchestration from the literature review, research project planning, and high-performance computing for the multi-job execution. Finally, you can use the Claude Science for the manuscript writing, the results documentation, and to reproduce your research work. Secondly, I believe that Biomedical Lab is built on the cloud. This isn't just a philosophical connection. Cloud is the foundation model that powers Biomedical Lab autonomous capabilities. And the 200K context window, the biological knowledge, the scientific reasoning, these are all from the fundamental layer of the cloud. Thirdly, the acceleration, I think, is real. And also the order of the magnitude is really exciting. 800 times faster on the specific biological tasks, and also 100 times for the overall biomedical science tasks. This is really to change what is feasible. Hypothesis, especially like the hypothesis that would have taken much longer time, can now be tested in like hours, right? Number 4 of the key takeaway is the gap between these 2 platforms. And it's getting, I think the gap are getting narrow, and both are expanding to each other's territory. So let's keep an eye on this kind of 2 AI platforms growth. 5th, I think you can try Biomedical Lab start from today because it's free, and if you have like the educational or the research institution emails, you can get a more computing source from the Biomedical Lab. This is no reason not to start experimenting the Biomedical Lab from now. Okay, I think that's all for today's lecture, and to compare the very promising Biomedical Lab versus Claude Science. And I think both AI platforms for biomedical science research are very important for your daily usages. I believe that this kind of a side-by-side comparison among the Biomedical Lab and the Claude Science will help you to find the right tools for your research work. Thank you, everyone, and looking forward to see you for the next video post. Thank you.